Good evening. Thank you all very much for joining us this evening. Here are the panelists. Can we- As you can see, they're very eager to be up on stage and to get started. I had the privilege and the honor to be able to spend some time with them just before this session, and what a delight! And I can tell you that you're all in for a real treat. What I'm going to do is introduce the panelists, and then each of the panelists will take a few minutes to give some background on the expertise that they have and the work that they have contributed to this topic on genes. And then we will open it up for discussion, for questions, answers. Okay? Okay, so to my immediate left is Dr. Stephen Scheer. Dr. Scheer is an award-winning genome scientist at Sick Kids and University of Toronto. He completed his PhD in the famous Sick Kids lab that identified the gene responsible for cystic fibrosis. Dr. Scheer contributed to the mapping and sequencing phase of the Human Genome Project, Chromosome 7, in the Craig Venter's Genome Sequencing Project. He identified gene copy number variation, or CNV, as a common form of genetic alteration in all of our DNA. And when CNV involves certain brain genes, leads to autism. Dr. Scheer published his first 2 scientific papers of over 600 as a Waterloo co-op student, and he's now one of the world's most highly cited scientists. To Dr. Scheer's left is Dr. Paul Boutros. Dr. Boutros completed his PhD and his MBA at the University of Toronto. He began his career in Canada at the Ontario Institute for Cancer Research and Medical Biophysics at the University of Toronto in 2008. 10 years later, in 2018, he left to be warm and has been at the University of California, Los Angeles since. In Canada, Dr. Boutros led our National Prostate Cancer Genomics Consortium. At UCLA, he leads an effort to create a genomic healthcare system across the state, trying to roll out genetic testing to every single patient at point of care. Dr. Boutros is passionately interested in the quality of genetic data analysis and created a multinational consortia to establish and drive adoptions of standards, particularly in research settings. He is a proud Waterloo alum, and he says, I quote, "Without co-op, I wouldn't be here." And to his left, at the other end of the stage, is Dr. Diane Williams. Dr. Diane Williams completed her PhD at Harvard MIT program in Speech and Hearing Bioscience and Technology, and prior to that, she completed a BSc in Electrical Engineering, also at MIT. In 2013, Dr. Williams started as a lecturer teaching courses in the biomedical determinants of health in the School of Public Health and Health Systems here at the University of Waterloo. In 2018, she assumed the position of Associate Director of the undergrad program in the School of Public Health and Health Systems. Professor Williams has a passion for teaching, mentoring undergraduate health science and public health students, and encouraging girls to pursue careers in STEM. She is fascinated by human development epigenetics and the promise of using genetic and epigenetic information and technology to better understand diagnosis, treatment, and prevention of diseases. She's also interested in ethical and social implications of this field. Okay, so what I'm going to do is turn the floor over to Dr. Scheer, who's going to provide about a 5-minute synopsis of his work.
Okay, thank you very much. I have a lot of material to cover in approximately, she said, about 5 minutes. So the title of this slide is A Brave New World in Genetics or Genomics. Genetics is a study of one gene at a time, and genomics is a study of all of the genes in your DNA in a single experiment. So you hear these terms come up through all of our presentations. And I showed this slide when I'm teaching, and I show it in different years. Now it's circa 2020, preparing for a time when genomes regularly show up in the clinic or your life. And that's actually happening now. In Toronto, in the teaching hospitals, there's over 20,000 different genetic tests delivered to patients each year, and that's increasing by 200% every single year. So this genetic information you're going to hear about, you'll hear a lot about autism and cancer in the presentation, but it's coming up for many different diseases. In fact, there's 6,000 different genetic diseases that, for which there's genetic tests currently available. So when you think about going to the doctor, what do they ask you? They ask you about your family history. They ask you to stick out your tongue and say, ah. And really, we would argue probably that in addition to environment, your genetics is the major factor that's involved in human disease. Every single disease has some type of a genetic risk factor involved. So the idea is that when you stick out your tongue and say, oh, they may take a swab or ask you to spit in the tube, and in fact, determine your DNA sequence before you leave the clinic to have that as part of your regular clinical workup. So now genetic information or genome sequencing is used to enable early diagnosis, and Dr. Boutros will talk about cancer, and I'll show you on my next slide, just touch on our work in autism spectrum disorder or autism. Genome surveillance is part of clinical visits for many different diseases, mainly for adult diseases like cancer. But in our hospital, at the Hospital for Sick Children, 70% of the patients that come to our hospital have some type of a genetic condition. And in fact, we're gearing up now that hopefully in 5 or 10 years, every single child that comes to the hospital will have their genome sequence done when they actually enter the clinic. But already we're seeing genetic testing in non-invasive prenatal testing, or NIPT. And this, essentially, the major genetic test now is to test fetal circulating DNA that's in the mom's blood. Okay, and it's tested using what's called chromosome microarray, or in some cases, whole genome sequencing. Pre-implantation genetic diagnosis is used to select for certain embryos to take forward into pregnancies. And there's over 2 million children born in North America each year that would have some type of a genetic test accompanied to pre-implantation genetic diagnosis. You'll hear a lot about stories now coming in the New York Times and Washington Post. I saw one recently about this concept of genetic risk scores. You've heard of companies like 23andMe and Ancestry.ca, where they develop a test or a statistical score of the likelihood you'll develop a particular disease based on a scan of about 1 million markers that touch on different pieces of your chromosomes. And now there's an even newer technology called CRISPR-Cas9, which allows you to edit or correct certain mutations. This is a revolutionary technology we're all using in the laboratories. It's only about a decade old, but in fact it's quite easy. And you probably remember in the news 2 Christmases ago where 2 children were born in China that were the first germline engineered babies that had been reported to be born using the CRISPR technology. So the brave new world is upon us. Everything you're going to hear about today is really is happening in the research environment, but in fact is starting to translate into clinical medicine. So it will impact your lives and certainly your children and your grandchildren's lives. The next slide is a summary slide of how we're using genetics to better inform on our studies of autism. I think most people know what autism is, but what they don't know is that 1 in 65 Canadians will receive a diagnosis of autism in their lifetime, and this number seems to be increasing over time. Our research lab has studied now roughly 3,000 families from Canada, where we've conducted a multi-million-dollar set of experiments and determined the entire whole genome sequence of 3,500 or so families from across the country. And there are families from the Kitchener-Waterloo area that are enrolled in this study. And now, using this advanced technology, we can answer the question that we're most asked by these families who participate in our studies, and that is, why does autism come about in my child? And for roughly 20% of the families using the whole genome sequencing test, we can now tell them that it is a genetic factor that is the result of that child's autism. And in this Toronto Star article, this is one of the first genetic alterations that was identified. In fact, it was discovered by Christian Marshall, who's mother-in-law and father-in-law in the audience with us tonight. This was based on a paper he was a first author on, and they're alumni of Waterloo. This is roughly found in about 1% of individuals that have autism. They have a missing piece of chromosome 16, a so-called copy number variant. But the technology now, as was said earlier, is way ahead of the current state. I mean, what we can do in the genetics lab is just unbelievable. And I just highlighted 2 examples. Okay, so for autism, we don't know, I shouldn't say, what I should say is there's no medicines for the core features involved in autism. And the reason for that is up until about a decade ago, we didn't have any of the genetic factors or targets that were identified to develop medicines toward. We now do for roughly 20% of the families. And there's a really revolutionary technology called induced pluripotent stem cell technology, where you can take a skin cell or a blood cell from a patient or an individual and grow it in the laboratory and then differentiate that simply by adding a few different growth factors into brain cells or heart cells or liver cells or lung cells. And it's critical for autism research because the cells that are involved in autism are brain cells, and you can't directly access brain cells of living individuals. So how do you test them? How do you test new medicines? Well, now you can take skin cells from an autistic individual, differentiate them in the lab, turn them into brain cells, and we do the experiments on brain cells growing in Petri plates in our laboratory. And we've done this now for many different individuals with autism. Secondly, you can use this CRISPR technology I showed on my last slide and make the same genetic alteration, or what we call mutation, in a mouse or a rat line. So we mimic in an animal model the same form of genetics that we see in the human patient. So we can take these animal models now and test through high throughput screening new drugs to see how they might modify and alter the developmental trajectory of these mice or these lines that we're growing in the laboratory. So we have these new tools that allow us to explore. I gave you one example of autism, and Paul will talk about cancer, but lots of other genetic diseases that you read about in the newspaper every day. And the critical question is not, can we sequence the genome? We can do that now. Can we study the disease? It's really how we're going to spend our money, what we're going to invest in, and how to take these things forward to benefit the families in the most effective way. Thank you very much.
Go ahead.
So I'll take this one sitting here. Is my mic on? Yes. Okay, excellent. Hard to tell. So maybe I'll start off in an entirely different direction than Steve just talked about. And I'll start off by telling you why I left and why I'm no longer called. So effectively, I was doing a whole lot of what Steve has just been talking about, but directed at a different disease, largely thinking about cancer, and cancer in particular that is diagnosed late onset, so in adults or in senior citizens, so people in the age range of 50 to 75. And all of those kinds of questions come up. Why do some people get the disease? What are the factors that predispose some of us to get a diagnosis and others not? And I spend a lot of time thinking about prostate cancer, which afflicts 1 in 7 men. So in a room like this, something like 10 or 15 of us will end up getting prostate cancer. Why 10 or 15? And why in some of those cases does it kill and some not? And we were starting to make some good progress in understanding the genetic factors. And we'd understood about half of those things that would lead somebody to either get it or lead it to be really lethal. But that still leaves 50% out. And it still leaves you the question, how do you translate that discovery from research into clinical practice? And so I was having dinner in a restaurant, and I got a call from somebody I knew at UCLA who said, you should really come out here and look. And I said, no, I don't want to go to LA. Why would I do that? And they said, well, okay, come out and we'll have a great dinner. And I was like, okay, that's a good argument. So I went out and I had the great dinner. And effectively, we talked about what do you need to start scaling this across a whole healthcare system. And there was a lot of optimism and enthusiasm about it. And so that's what the last 15 months of my life and the next 5 years are really going to be consumed by. What we're doing is thinking about how can we make sure that every single patient that comes into a statewide healthcare system with nodes in each of the major cities in California, public, able to have the same quality of care independent of where you are, and getting a genetic test as part of it. And so working with my colleague, Arash Naeem, who's a geriatric oncologist, we started to think about how can you actually make sure patients understand that? How do we make it a routine part of the clinical workflow? And so what we've set up is an iPad video consent process. So every time a patient walks into a clinic, they get an iPad. The iPad gives them a 30-second introduction to the idea of what does it mean to be consented for a test? What does it mean to give your approval to have these types of things done? And then 4.5 minutes talking about the consequences and ways in which genetic testing could be done. At the end of it, they go ahead and click on a consent that allows them to, can I get the next slide? Maybe. There we go. That allows them to go and agree or not agree to be able to do this type of work. It's in 7 languages. There's core translations in Spanish because 50% of the LA region is Hispanic, and English, but then there are captions in a set of other languages. It's only for adults today. It's much more complicated to consent and assent for pediatric studies. And in particular, we want to make sure that there's an opportunity for both the children to assent to the testing and for the parents to give consent for it to be done for them. And that requires a bit of special hand-holding on that to get it right. But in total then, we're able to start making sure that people see this iPad as soon as they go into the hospital and see the receptionist. And as soon as they're finished, it gets a wipe down with like a Kim wipe to make sure that we make sure it's not going to spread contamination, and then goes to the next person. And that allows us to be rolling out something on the order of 2,000 patients a week. We did 2,000 last week. And we're not going ahead and saying, okay, well, we want to do a genetic test, so go and get a phlebotomist and do a specialized blood draw and anything. Instead, we wait until there's a routine clinically indicated blood test, and we use the remnant blood from that, which today would just be thrown out. And instead, we use that. We have a 3D barcode that ships it to a pathology lab that does a DNA extraction, and then we use that to do the genotyping on those patients, and it flows directly back into the clinical record. Can I get the next slide? So ultimately, what patients are consenting for are 3 things: to allow us to use any remnant samples of blood or tissue for research purposes, to allow us to do the clinical genetics assay to figure out whether or not there are any actionable variants, anything that might change care in their genome, and to link all of that to de-identified dumps of their full medical record for research purposes. We think this year we'll have 150,000 patients rolled out, which sounds good. The UCLA health system is 5 million patients, so it's going to take us a while to scale it. And today we're seeing about 70% consent rates. That's not fantastic, but that's actually pretty reasonable. And maybe most encouragingly, the consent rates are not biased by age or ancestry or sex or any socioeconomic feature that we've looked at. So we think that this is one path to start to rolling it into the whole healthcare system. And I'm looking forward to talking to you guys about that.
Go ahead.
Okay, sure. So I teach students in the School of Public Health, students in health sciences and public health, who are really interested in the idea of prevention of complex diseases as well. And so if we think about diseases like diabetes, cancer, obesity, mental illness, substance abuse, all of these are going to have causal factors that include genetic factors as well as environmental factors. So I teach a course on epigenetics and health, and epigenetics is really that way of bringing the genes in together with the environment to think about how they interact to increase risk for diseases over the lifespan. I also teach several courses on prenatal development, and if you think about it, of course, we all started life like the cell we see here in the figure, that single zygote that has the genetic material from mom and dad that has the potential to form any cell in the body. And I've always been fascinated by human development, how we all start out the same way, and then you have to develop into these different cells and different tissue types, and that's just through differentiation of different cell types. So cells need to express certain genes from the DNA to become, say, a liver cell or a pancreatic cell or a neuron, for example. And one of the ways that that process is controlled is through what are called epigenetic chemical modifications of the genome. So it's not actually modifying or changing the DNA code itself, but it's actually chemical tags that are placed on the DNA directly, such as what are called methyl tags, or changes that affect the actual structure of the chromatin, which is how the DNA is organized. It's either tightly wound up, and if it's tightly wound up, then you won't be able to express those genes, whereas if it's relatively loose, you can express those genes. So this is a process that unfolds during development that affects cells differentiating into certain cell types in different tissues. But what's really interesting is where the environmental factors can come into play. Because we all know, for example, with a complex disease like cancer, there's certainly a huge genetic component to that. We have mutations in genes that affect the cell cycle. But then that interacts with environmental exposures, such as whether you eat a healthy diet and exercise, or whether you expose yourself to potential carcinogens like cigarette smoke. And that these environmental exposures may then come in and change some of these chemical modifications and affect long-term gene expression. So this certainly could happen during prenatal development if we think about prenatal exposures, whether that's, say, a mother who drank water that was contaminated with lead, or say, a mother who smokes during pregnancy. Or it could even be through animal studies. It's been shown that even the dad can contribute through their exposures, environmental exposures, that could contribute to changing these epigenetic markers that could increase risk for diseases. So from a public health perspective, we're really excited about the idea of thinking about prevention at the level of figuring out what are these environmental exposures, how do they interact with the genetics to affect gene expression. And the idea that here is something where you could maybe have some reversibility. If you knew how these chemical tags are affected by environment, maybe you could reverse that. Certainly healthy lifestyle like exercise may be one of those factors that reverses some of these adverse epigenetic modifications. So our students are really interested in this idea of prevention of disease, and that maybe epigenetics could be a way to reverse a disease process.
That's great, thank you. Okay, I know you're all sitting there with lots, brimming with lots of questions in your minds, but for some of you that do not have a question in mind, maybe what I'll do is I'll start with a question of my own for each of our panelists, and then we'll see if you're ready to ask your questions. Okay, so I'll start with Dr. Scheer. I was an older mother. I was in my late 30s when I was pregnant with our first and then a set of twins. So my husband and I were debating. We had a really difficult time. We struggled whether or not I should get amniocentesis. In the end, we did. And but I have to say, even to this day, it was very traumatic. To this day, I cannot drink bubble tea without thinking about the needle that was the size of it seemed the size of the straws that you use in bubble tea.
That's
that
big.
went in, and then with the twins into each of the yes. I know you're cringing. Are you telling me now that prenatal tests are no longer have to be as invasive?
Yes.
Would you care to elaborate?
So yeah, most advanced maternal age testing now is non-invasive prenatal testing, like I mentioned. So not all of it. It depends on the situation, but the majority is these cells from the fetus that cross the placental barrier and go into the moms. And there's special ways to isolate that DNA. It's based on the size and how it fragments and things. And in fact, yeah, most tests in Ontario now, and actually around North America and Europe are based, actually around the world. And they test for the same aneuploidies, the chromosome number changes, the large changes, and now they're looking more specifically at some of these copy number variants that my lab helped discover. And then ultimately, there is a lab in California that does 24-hour whole genome sequencing now. So if there's a family history of a particular condition, that's a test they can perform. So it's still early days, but certainly for the chromosome numerical changes like trisomy 21 and chromosome 18 changes, the standard test is not as invasive as it used to be.
That's wonderful, Dr.
So that's good and bad. It's the numbers are significant. I also wanted to point out that I had my first child when I was 40 years old, actually. So this is a phenomenon in the Western society that many of the disorders, diseases, conditions, depending on the terminology you want to use, are a result of our change in environment. And one of the major changes in our environment is the age at which we have our kids. Our biology, as in the human species, evolved around maximizing our health, of childbearing health, around a much younger age. So we've ourselves have changed our environment. In this
So
case.
are you saying it's not always the women's age, that
Right.
the father's age
Yeah,
also
so as
has
we were
an
talking
impact?
before, one of the somewhat surprising findings, although in retrospect not surprising, is I didn't mention it, but the number one factor involved in developing autism, called a risk factor. It's not a great term to use, but that's the technical term we use, is if you're a male or a female. It's a 4-to-1 male to female ratio. And secondly, there's a 2 to 3% added likelihood if you're a male over 35 years old, because your sperm, when they're replicating as you get older and older, make more mistakes, and you get mutations accumulate, not just for things like autism, but really across the board. Because, again, as a species, we were primed to have our kids much earlier. We've changed that, so as we're seeing different effects in the clinic now.
Thank you. Dr. Boutros, did you want to add to that before?
I would add that increase in non-invasive testing is influencing almost every aspect of healthcare now. It's really an encouraging change. And so if you take an area I know well, prostate cancer, increasingly diagnostics are being done just on urine. So we no longer even need to do a blood test, never mind sticking a needle in. And those tests are slowly receiving the late-stage clinical trials and moving into routine practice. But it's easy to imagine in a few years that the amount of invasive testing done in all diseases will drop dramatically because of these technologies.
Could I just add one comment to this? Because I know there's a lot of techies in the audience, particularly being from Waterloo. What we've, as a community of scientists, did for the Human Genome Project is sequence a reference genome. And that cost roughly $3 billion. And the first identified individual had their genome sequenced with this Craig Venter and James Watson around the same time was $70 million a genome. Now we're at about $1,000 for a whole genome sequence. Some of these panel tests that Paul would do are $20 to sequence all of the cancer genes. Maybe not 20, but $100 is coming down. Okay, so really the heel prick test that you have now, as your baby would have, will turn into a whole genome sequence test very soon once the prices plummet. So you can do this. Technically, there's a start and an end point. The data is finite. You get it. You can capture it. It's a lot of data, but it's actually not that much data when you think about it. So the technology here, what I wanted to say is now we can actually use the same technology that we use to sample blood to sample single cells. Okay? And I know there's biology professors in the audience who are doing this here too. It's really unbelievable. So if you sample a tumor, you take the whole tumor, maybe only 30% of it is actually cancerous. But if you can get down to the single cell that's cancerous, you get a whole new picture of what that cancer looks like. And I think much of biology will be resolved at the single cell level. It's really incredible what you can do. I'll just throw it to really get you hyped about the technology, because I didn't have time when I was up there. Okay. You can sequence, DNA is pretty stable. In fact, the oldest DNA fragment that's been sequenced is from a 700,000-year-old horse that was found, somewhere in Europe. 700,000, and the entire genome of Neanderthals, our predecessor, a human species we cohabitated Europe with probably 80,000 years ago, has been sequenced. You can sequence DNA from pretty much anything. In fact, most people don't know that Caucasians, European ancestry, share 10% of their DNA roughly is actually Neanderthal. Neanderthal and Homo sapiens made it. You can sequence, I tell my students, the experiments they do are only limited by their creativity now. You can read, write, and edit and create your own organisms if you really want to.
Right. Wow.
So I'll comment on the amount of data there. In some sense, I agree entirely that it's not that much data, but a human genome, the way that's done today, is about 3 billion bases. So that's a lot of measurements. Most of it doesn't change, so it's not really that bad. You measure each of them 30 or 50 times. So that's 50 times 3 billion. And then if you're doing one of these single-cell sequencing experiments, then you do 20,000 cells on top of it. You suddenly start to get gigantic amounts of data. And it's fascinating because the biggest problem in data isn't the compute power and the processing, it's storing it. And there are cases where it's clearly cheaper to just throw something out and say, if I need it again, I'll sequence it again, than it is to actually keep the raw data. Obviously, we'd never do that for patient data, but it starts to get you down the road of just how fast the price of sequencing has dropped, that it even outshadows our ability to store the data.
That's true.
Yeah.
Fascinating. Boy,
Well, some of you will know we have a collaboration on our big autism project with Google. And when we transferred the first tranche of data in 2015, it was apparently the largest data set they had received. Now, and Paul's right, it actually costs us more to download the data than to generate the sequence. There's a change in time, but what I was trying to say is, for your own genome, just a linear data set you can carry around on, a USB key. But you're right, when you calculate and compare and all this, you have to have large supercomputing, cloud-based infrastructure.
Dr. Boutros, I have a question for you related to just what there is for us to do. Is our health predicted by our genes, or is there anything else we can do? Like, say I develop cancer, is it worthwhile even, or do I give up? Is it worthwhile looking at other factors that can influence the progression of a particular disease?
Yeah, it's a great question. I think some parts of our health are very directly determined by our genetics. There are certain disorders that are determined by just a single base out of those 3 billion. But most of the things that we think about, especially once we get to adulthood, are complex. They're the interaction of our specific genetics with the way in which we live our life through years, right? There's a lot of evidence that the earliest beginnings of a cancer will be 20 or 30 years before somebody is diagnosed with it, which means that lifestyle is an incredibly powerful factor for us, and we should never forget it. And maybe something to think about is we actually have no medical intervention that's as broadly effective, cheap, good cost benefit as exercising. It is like, it's distressingly how good it is. A great example of it is, let's pretend that you take a cancer patient and they've newly been diagnosed. If they start an exercise program, independent of their level of fitness, independent of anything else, they just increase by about 20 to 30% what they're already doing. It has as big an effect as the best chemotherapies that we have. And that's an exercise program. And actually, we're doing a lot of, so one of the nice things about being at UCLA is that it's got a fantastic sports science program, and it's one of the main Olympic athlete training places. And so we're now trying to turn that apparatus to think about health problems. And so we're doing clinical trials where we look at samples from cancer at a patient who is newly diagnosed, and then have them go through an exercise program for 6 weeks and see how that actually changes the tumor and changes outcomes. So, and of course you can come up with lots of other effects, but exercise is by far the biggest one. So yeah, there's a lot that we can do to moderate these things.
That's right.
It's cool. Now, is there the idea of being able to monitor how that tumor is progressing as they're exercising? What would you be looking at in that? Would you be looking at epigenetic factors, or would you be looking at genetic factors, or just how the tumor is growing when somebody's, say, exercising or doing something to improve their outcome?
So when we think about these things, we always want the hardest outcome. And here the hardest outcome is talking about how solidly related it is to patient well-being. And so we'd really love to be able to link exercise to overall survival of the patients, or to the likelihood that their tumor metastasizes or advances. And there have been a lot of studies that look at large cohorts of people to be able to nail that down. When you get to the more targeted molecular studies like we're doing, the two best things to look at are probably, for cancer, evolution. How many types of cancer cells are there? And that you can do, just as Steve mentioned, by looking at the blood and seeing how complex the set of mutations and DNA types you see in the blood are. And you hope to see that reduce over time, because that would be indicating that some parts of the tumor are dying out and can no longer live properly.
Yeah.
That would be the best molecular. Of course, we also image the patients and look to see how the tumors are changing and look at other things.
Yeah.
So one comment on, just to reemphasize what was said earlier, your genome or your DNA, it's a blueprint for your life. And a blueprint is a really good term because sometimes it's entirely predictive, like mutations in the cystic fibrosis gene lead to cystic fibrosis, but most cases it's a blueprint. You can use it as you choose. You can modify the outcomes based on changing your environment. But what's been surprising to me over my career since I was a graduate here is for things that we didn't think were genetic, we're finding out they're partially genetic, or in some cases entirely genetic. We really are pretty hardwired. At the same time, things we thought were entirely genetic, we're seeing that environment or modifying. There are other components or other genes involved or other. So it's the yin and yang. It's black and white is 50. I always go, how much is nature, how much is nurture? And at the end of the day, it'll be 50-50. We did the experiment. It was called the Canadian Personal Genome Project. Some of you may have read about it in the Globe and Mail a few years ago. So we collected DNA from 100 healthy volunteers and did whole genome sequencing. And then asked the question, based on the state of the knowledge, how many would have benefited from prediction of their mutations they carried. And about a quarter of the participants, we found one of these, a mutation there's a list of genes called the American College of Medical Genetics 59. There's 59 genes in your genome that if you have a mutation, you will develop a defined disease, and there's actually a clinical intervention, either a medicine you could take or you can have surgery. A lot of them are cancer genes. So about a quarter of the people carried one of these mutations or so. So actually getting that data would have been, many of them already knew they had a family history, but it was informative for them. Every single one of those people was carrying a mutation in a recessive gene. So had they married someone else who was carrying, like cystic fibrosis is a recessive disease, thalassemia. So that information can be quite informative, in particular in populations where there's a high rate of first cousin marriages, consanguinity, like in the Middle East, 20-30% in some countries are first cousin marriages or more. So recessive disease, but what's really striking is every single person carried at least 3 mutations in genes that we know interact with known drugs you take, so-called pharmacogenetic genes. It's not a lot of data how to use this yet. It's still very rudimentary, but we know that these mutations will inform on the types of drugs and the dosage of drugs you'll take once the databases of information like Paul's collecting from healthcare systems have accumulated. So the data, it will be used now that it's pretty easy to get the information and it's relatively cheap. But it's really just the beginning. It's the tip of the iceberg. We don't know much yet. We think we know a lot, but we think as we sequence entire populations. I don't want to take too much of the time, but just again so people know, in the United Kingdom, they in their National Health Services they've sequenced now 100,000 whole genomes. They've got money to do 5 million. So everybody in the National Health Service will have their whole genome done. So essentially anybody who goes to the hospital. And they're sequencing through the UK Biobank project 1 million people. In the United States, they're doing 1 million through it's called the Olivis program. You'll hear a lot about this. And in Canada, we're trying to have a population sequencing cohort. So we see what the gene collection looks like in the general population. Most of our data is from cancer or autism or inflammatory bowel disease or whatever.
Fascinating.
Yeah.
So, Dr. Williams, knowing all of this, and that this is just the tip of the iceberg, and it's the number of tests that can be done in genome and genomes that can be, the data that can be collected, I would think if I were one of these research or participants or patients, I would want some counseling, or I would want to have access. Is that a career path for some of our students?
I think it's going to be a growing career path because I think most people in the general population are really not good at understanding risk and how to take that into account. Even those people who've done, say, one of those 23andMe tests where you get all this information, and most people wouldn't really know how to interpret that information or how to make lifestyle choices. Even just remembering I was considered advanced maternal age at the age of 34 when I was pregnant with my first child, and I went to see a genetic counselor, and she talked to me about risk, and I'm kind of a mathematical person, so I'm like, yeah, I get it, I understand risk, and I decided I didn't need the amniocentesis because based on the number she gave me, I knew that the risk for my son having aneuploidy was low. But I have that mathematical background. Most people don't know how to interpret risk and may kind of panic about information that they get. So you really do need somebody who can teach people. Genetic counselors know how to talk to people and explain risk in an easy way that they can understand. And so I see this as a field that has potential for huge growth, not only with respect to people getting genetic information from clinics, but also from some of these companies where you can just send along some of your cheek cells in a test tube and they'll tell you information about your genome. So yeah, I definitely see genetic counseling as being an important part of the picture to help people understand the risks that they have for different diseases when they get this genetic information.
And then
It's
how
worth-
complicated are these roles now? It's not just assessing risks, but once the test is done, what do counselors do after that with that information?
Yeah. Well, I mean, part of that
Anyone?
is, they can't tell you what to do, and I think that's the issue, is they tell you what your risks are, and you have to make the decision. I had to make a decision, do I want to do an invasive test, like an amnio or not, and my husband and I were comfortable with us not doing it. And I guess this is the same thing as going to your doctor. They tell you about your risks and whether you should get certain screenings, and it's up to you to decide. But I think the counselors play a really important role in educating people.
It's worth saying our clinicians do not get enough education in probability and risks and communicating that today. And if one was to take as a relatively selected population cancer clinical trial clinicians who you'd hope would have a good sense of risks and give them what to somebody who does this every day is a relatively simple calculation, they will not interpret it correctly. And that makes sense, right? They're focused on different things. And so genetic counselors, I view, play a critical role in also taking some of that burden off of frontline clinicians who need to think about other things.
It doesn't make sense to me because genetics is the basis for medicine, and most medical schools, in your 4 years of medicine, you get two weeks of genetics. So most of your genetic information comes from your undergrad professors right now. That's changing in curriculum. Genetic counselors it's a 2-year MS master's program. And we were talking before, in Ontario, U of T is the only school, and there's only 6 new students a year. If you have kids going, this is a growth area, so it's a good, but it's very hard to get into, very, very hard. But what I wanted to say is the testing results that come back from 23andMe, and I've heard there's close to 1 million Canadians who have used either 23andMe or Ancestry. And Ancestry now does genetic gene mutations too, not just Ancestry. Anyways, you don't get that data back through a counselor. But anything, if you're, any hospital-based testing here in the province, you would get it through a genetic counselor or a clinical geneticist. It's an incredible program. So I had my genome done probably first, very early on, and I thought I knew a lot. And when you look at your data, I had to use genetic counselors too. So I think that's the strongest recommendation is if you're doing this kind of testing, make sure you're getting it back in the most informed way. And the counselors are trained to do that. They deliver it in the right way. And when you have your testing done, it's only good for a limited amount of time. And so, actually, again, Christian Marshall, your son-in-laws are good. Not always bad like me. No, but anyways, so he did a very nice study of sick kids. It was a whole genome sequencing study, and took 100 kids who came through clinical genetics as sick kids who did not have a molecular diagnosis yet. So they were so-called idiopathic or uncharacterized. Did whole genome sequencing, and for 25% of them, we could find a genetic diagnosis just in a simple analysis. Take the same data, because the genome is essentially static. It's what you get at your birth, and it stays. So once you have the sequence, that's the beauty of it. It's a scaffold you can use to interpret your blueprint. Went back and looked at it 18 months later, because there's a lot more data to compare and contrast against in databases, 33%. So it went up 8%. Looked a year later, it went up another, I don't know, 5 or 6%. So it's all a numbers game. Genetics is statistics. The field of statistics actually grew out of human genetics, out of genetics, believe it or not. You know, it's the story I usually tell. It's like early global positioning GPSs. You remember when you first got your first GPS in your car, and it sent you the wrong way every time because it's like Google Maps. Didn't have enough data to compare against. We're early GPS days in the genome sequences. Once we have millions to compare against, it's going to get pretty good. It comes back to how much you believe you're hardwired or not. When we have millions to compare against, using AI, machine learning approaches, you'll be able to decode the evolutionary decision tree. That's what I think about a lot. But we need to get into the millions and have broad populations of sequences. It will come. Do you agree with me?
Oh, absolutely.
Yeah.
Yeah.
Wonderful.
The only place I disagree with you is I've not had my genome sequenced and have no intention of
You
it,
don't?
which is
Okay.
a fascinating contrast.
Well, there you go.
I'm trying to
Okay.
decide now whether to do it or not.
Me too. So this is wonderful. I don't want to monopolize the conversation, so we're going to open up the floor to, I think, do we have mics to, or is the acoustics good enough for, so in the back, yes, please go ahead.
I was wondering if, can you hear me?
Yeah, it's really good, yeah.
I was wondering if you could chat a bit about how environmental stress factors, whether that be financial status, education, lifestyle, whatever it be, ties into
I promise that noise isn't me.
Yeah,
I'll start
I'm not-
off in... I don't know why I felt the need to clarify that. I'll start off in cancer because it might be the strongest effect in cancer, and then I'll pass it off to my colleagues. So ultimately, your genome is your genome. It's you know, the blueprint of what you've got, and then you build on top of it. But stress and a whole series of lifestyle factors change the way your body interacts and responds to different things. And that's really particularly clear in cancer, where we can actually take a look at the mutations that accumulate on top of your blueprint and figure out today what were the specific stresses that led that cancer to form. And we can say, oh, about half of the causes of this cancer appear to have been smoking, and about a third is random stochastic effects of age, and another sixth or whatever I left out of that is some other factor. And you can start to draw those maps out just by looking at the types of mutations that happen. And you know, I think it's clearly the place where we have the best chance to modify and moderate things. In general, a universal statement is that stress, and in particular inflammation caused by stress, is really bad in the context of cancer. It's one of the most critical underlying factors that predispose everybody to get it. And you know, if you think about why I mentioned exercise, exercise reduces inflammation, reduces stress. It's probably one of the major mechanisms by which it reduces the rates of cancer. And you can make the same comment about, oh, you've exercised, therefore your chances of cardiovascular disease are lower, therefore maybe you're less stressed about other things. So it's really a systematic factor that affects effectively all of human health.
Yeah, I mean, we could talk for hours about this. You know, I think things like mental health, in many cases, they link or relate to things like stress, sleep. And there's classic studies that have been done that are, I think, just groundbreaking, that have shown that, you know, when you're a child, they've been done in mice and rats, but also in humans now, the amount of physical interaction, including the breastfeeding, can, through epigenetic processes, influences significantly the genes that are turned on and off, right, that influence how you develop. So in the same way, stressors can impact your mental health through the same mechanisms, actually, in fact, the same genes. So it all plays into this, this kind of process, I think, moderated through this Rosetta Stone we call epigenetics, but it does link back to the DNA. And that's why I argue over and over again, to make sense of any of this type of more complex gene transcriptional data epigenetics, if you have the whole genome sequence, that's your scaffold of information. So that's what we're arguing for our studies, that you just need to do that once and put it in the database and then try to map all your health record information. That'll be the best way to get to this issue of nature and nurture, genes and environment, and how to benefit that in your health decisions. But I think the one thing to keep an eye on, if you're a student thinking about graduate school, most of what we know about cellular biology right now has been based on our knowledge of studying cancer genes over the last 30 years, because that's where all of the research money has come from. But in fact, the exact same processes, but probably with different genes, will be found in both mental health or neuropsychiatric conditions. We learn a lot from the cancer field. There's somatic mutations in all kinds of things that we didn't think were genetic before. So schizophrenia, obsessive compulsive, all these different, and autism I talked a little about, even cerebral palsy and others. So that's the field I would go into if I was a student. And stay away from cancer, that's already been done. The next round of Nobel Prizes are in studying neurodevelopmental neuropsychiatric conditions.
And I'd echo something Steve said in his actual presentation before, which is this idea that we can make a cell of any origin transforms research in other fields. Another reason cancer research moved so far ahead was because cancer cells like growing. So you take them out of the body and you put them somewhere and they keep growing. So you could do experiments and they were tractable. A lot of other human cells didn't have that characteristic. So the lack of a model in which to do experiments was really challenging. And over the last 5 years, that's really transformed experimental biology.
Yeah, it's true. Like the studies that you're doing in your lab, Stephen, with the induced pluripotent stem cells, that you can get them to differentiate into neuron-like cells and then kind of study the brain in a dish. The idea that you can look at, some of the epigenetic or genetic factors in those cells and look at drug treatments and how they affect the cells, in a dish.
Yeah, I'll just comment because you introduced, when I was a PhD student, it was 30 years ago, in 1989, the cystic fibrosis gene was identified. To be honest, I remember at the time the announcements, there'll be a drug that comes, and waited and waited and waited. 30 years later now, they're actually 90% of patients will be treated with a complex of drugs, 3 drugs together. It's a cocktail. How effective that is, we'll see, but certainly for a proportion of them, they're being treated. I just raise this because traditionally it takes about 20 to 30 years to go from a gene to an effective treatment. It takes a lot of time and investment. With these new experimental technologies and high throughput screening, we can do things a lot faster. And there's some rare genetic diseases, again, you're reading in the paper, things like spinal muscular atrophy. There are drugs now that cure these diseases, and we've got some new ones coming that people are working on sick kids. There's a clinical trial using CRISPR in types of muscular dystrophy where it's working very well. So again, you're only limited by the creativity and the money. The biggest challenge we have in the field now is the drugs that are coming from these new technologies are hugely expensive. So the spinal muscular atrophy treatment is $500,000 a year. Who's going to pay for that? CF is probably around the same pricing. So I think as experimentalists, in society, we have to decide what's important and what's not. These companies, maybe they don't need to make billions and millions of dollars. But the thing I think about most is we will develop drugs for autism at some point in the future, and thinking a lot about developing business models to keep the prices down. Otherwise, we're going to end up in the same place.
Yes. That is such an important message, and such a personal one for me, because I, as a young teenager, worked in Easter Seals camps with cystic fibrosis, children with CF, and giving them treatment several times a day, and was just heartbreaking knowing that after I left camp, some of them wouldn't be alive in a few years. So this is so promising, and of course the cost of drug, and in autism we're so many families are devastated and affected by that. There was a question here. Yes, please go ahead.
This has been great listening to all of you speak. It's been very interesting. So thank you for coming here. And I hope my question isn't too invasive, but I was wondering why so many of you seem to be held back from having your own genome sequenced. I hope that isn't too
So I'll start off and give mine, and then Steve already has had his, so he can explain what the motivation there was, I guess. For me, I'm fortunate. I am young and healthy, and I don't believe that my genome is going to tell me something, knowing my family history, that I don't already know, which is that I should eat healthy and go to the gym and not be too stressed and sleep well. So I kind of feel like I know those things, and at this point in time, given what we know about it, it is not going to transform my personal life, one. Two, it's going to get cheaper in X years, so why would I spend X dollars for it today when I can spend 1/10 or 1/100th of that later? Before long, at some point in time, I won't be healthy, and at that point in time, I think it most likely that it'll end up getting done in a healthcare system, in which case that's even better because it'll directly flow to caregivers and those sorts of things. And then lastly, it probably runs in the family. None of my family members have chosen to do it for all effectively the same reason. And, I'm very lucky to have, parents who live into their 90s and grandparents who lived into their 90s and stuff. And so I kind of go, well, I'm not stressed about it. So that's what it was for me. So it's not an ethical or deep personal concern. It was, I just didn't believe in my situation it would change my life.
Yeah.
You've been thinking about this.
Well, now I have been thinking about it, but maybe I won't, because same thing. I'm the youngest of 8 kids in my family, so I can just look to my older siblings and see what I'm going to have to deal with. So I know, same thing, I know my family history. Irish Catholic parents. But anyway, I kind of know what's coming up, and I know what my family history is. I know high cholesterol runs in my family, so my doctor's really good about getting me tested, and so far so good. I've been lucky, but none of my siblings have developed cancer, and some of them are into their 60s now. So it's kind of the same reason, knowing my family history, would I really get any more information from a genetic test? And would I change my lifestyle in terms of, I know I need to eat right and exercise anyway, and keep the stress down and sleep,
So maybe I can.
Your turn to convince.
Yeah. You could talk. How much time? How many people in the audience have had 23andMe or a microarray done? Put up your hands. So I'm seeing about a half dozen or so, or maybe a dozen. The genome sequence, probably no one has had the whole genome done. But with the microarray, you get 1 million to 2 million data points, and for common diseases, you can impute that across the genome. It's actually a really good test, and you get the ancestry. That's what most people want. Genetics is the second most popular hobby in the world, I've been told.
Really?
Gardening is the first. So, I mean, this is really what they're selling. There's been millions of these tests done. It's incredible. But anyways, the whole genome sequence for me, Dean Sass, who was one of my co-op students in the audience here, 1992? 95. You may remember 999-1 DNA sample. That was mine because we needed to have a DNA. Whenever we got new technology, we needed to test on someone, so an obvious person would be me. But in fact, for the Personal Genome Project, I didn't do it as part of that project because I funded it from a philanthropic grant. I didn't want to be seen as benefiting directly. They were volunteers, a lot of genetic counselors actually, and professors of genetics and things. But anyways, yeah, if you're healthy and you don't suspect there's anything wrong, there's no reason to have it done right now. I agree with Paul. When there'll be an inflection point when the price comes down in the $100 range, and it'll be ubiquitous in the healthcare system. It's just another form of family history. It'll be really interesting to see. I think they're quite complementary. The picture I showed of the doctor sticking out the tongue. I think if you have an adopted child, you should have the genome sequence done. But anyways, as the prices come down, the information you get from a verbal family history will be very complementary to what you get from the actual genetic sequence. Eventually, they'll merge as we get more and more data to compare against. But ideally, you really do need both. It's different types of information. So all of these projects now are trying to couple genome sequences of some form or another to the electronic health records and in existing databases. In Ontario, we have this Institute for Clinical and Evaluative Sciences that has captured a lot of this health admin data, and we're trying to link through different projects. It's the future. You'll be using the data, in the next generation.
First of all, I'd like to say a huge thank you to the Hospital for Sick Children in Toronto. And 40 years ago, my oldest daughter was diagnosed with spinal muscular atrophy. And she's an undergrad of the University of Waterloo and got a master's here and is employed with the city of Waterloo and has done very well. And I've heard about this wonder drug that is so expensive, but I think it's only for very young children, under the age of 2, 1 or 2 or something like that. So hope for the future, but anything to get the prices down, to do some treatment early would be wonderful.
Yeah, can I just jump in on that? Because so people know, it's in the newspaper almost every week. I don't know the exact numbers. There's about a dozen kids who are being treated, I think, in Canada. And it's paid for by philanthropic donations, like people like yourselves too, the sick kids and that, right now. Or out of hospital budgets that largely kind of just scavenge money from other places. But I'll make sure your comments go back. You know, the research really provides hope to families. In some cases, it provides cures and treatments right away. Most cases, it takes longer. Usually impacts the next generation. One thing I wanted to, just add
no, that's
to come back to the, surprised people haven't asked, but it's kind of related to the last question. In Canada, we have what's called the Genetics Non-Discrimination Act as of 2017. And in fact, it's really worth acknowledging Bev Heim-Myers, who's a Kitchener-Waterloo resident, who led that push. Some of her colleagues are here. And it protects against genetic information being used by the government or insurance agencies or in an adverse way against people. So this is an incredible thing that we have in Canada. As far as I know, since that act became law, there's been no negative scenarios that we've heard of. So when you have this testing done, so the answer to your question usually in pre-2017 was people could use the information against you in some way. They can't in Canada anymore. So that can't be, it shouldn't be an excuse anymore. But it was an excuse. There are a lot of families coming to sick kids. As I said earlier, we have strong genetics department because 70% of the kids who come there have genetic testing done. There were families who were not having specific tests that would have benefited their kids because they were worried about insurance acts and things like this. So that's changed now. So that's a good thing.
Thank you. Thank you for bringing that up because it touches a little bit on the ethical, legal issues. Maybe we have time for one last question. Please go ahead. Okay, 2 last questions.
So Dr. Boutros, you mentioned that you have about a 70% uptake on the consent form. What are the things in the 30% that tend to keep people from signing that form?
So we've done a lot of work on that, as you might imagine. The number one factor is the severity of the health condition that they are being seen for at that time. So effectively, if the patient has cancer or something that is potentially life-threatening, the consent rate is nearly 95%. If they are coming in for a dermatology appointment, the consent rate is quite a bit lower. And so effectively, and those are just examples, but effectively, that appears to be the driver. And we're actually instigating, if you did not consent, can we ask you again in the future? And so in a pilot, it's 100%, where I think people are basically saying, not no, but not now.
Exactly.
And so we're going to need to carefully distinguish that in the system. So it's a great question.
And most,
I anticipate that when somebody is in that setting, sometimes they don't want one more thing to think about, and the defensive reaction is, I've got a lot on my mind, and I got to do this, and I'm going to work after, and blah, and is this going to immediately benefit me when I am here to get a renewal of a prescription on a skin cream, or to get a tag removed, or something like that? People's default reaction is, no, I don't know if I understand the benefits to me. I don't think there are risks, but maybe there are that I'm not thinking about. Let me default say no. And that's our best understanding of that population. Yeah. We're actually going to be doing a set of qualitative interviews on the no population moving forward as well to get a better sense of it. But so far, that's what the data shows.
That's exactly what we've seen also in many different genetic studies is usually they're invited into the study at the time of getting the diagnosis, and they just have these consent forms, or they don't want anything to do with it. They have to go deal with their life. Usually they're busy, but they come back. No, but that used to be the issue. Not anymore. I have to say that we've had, we've done thousands of families from many different studies, and whenever we go back and reconsent for another added study, we have a roughly 90% uptake, and we've only had a few families drop out. And often when they do drop out, what happens is the children in our study become age of assent where they make the decision. In fact, we were talking earlier, I can't remember if it's 15 or 16. I think it's 15 years old as sick kids in our studies where the child or adolescent would take over actually their data if they wanted at 15. So they can drop out of studies if they decide to. But typically Canadians are very supportive because it's a socialized healthcare. So it's not the same in most places in the States. California is different. This is one of the reasons that there's been so many productive genetic studies done in Canada.
Very good. Okay, thank you.
Hi. So there's often a lot of conversation about reversing epigenetic changes, such as reversing methylation. Do you think there's any conversations or any research going on now or in the near future about inducing methylation or inducing epigenetic changes, like methylating oncogenes or methylating apoptotic genes for things like Parkinson's?
I think most of the studies have been in animals where they've been able to treat them with drugs to try to remove methyl tags. The issue is that it's very non-specific. So you're treating with a drug that inhibits an enzyme that would tend to add methyl tags, and that's just across the board. And so because you can't be very specific, you don't know there could be certainly adverse consequences. You might think, well, it'd be a good thing to remove, say, methyl tags from tumor suppressor genes so that they can be expressed, but you could be also affecting other genes, say oncogenes, in the opposite direction. So I think that's one of the issues is it's non-specific. What I'm always interested in, what are the things that you could be doing naturally to reverse epigenetic tags, and that's still not a very well-researched area. For example, does exercise take off some of these methyl tags, for example, and what would be the mechanism for that? It's really not understood what is controlling when tags are put on and when they're taken off in a very specific way. So there's still a lot of unknowns in the field, I would say.
I'll add two things to that. It's a great question. One is that there's this concept of the epigenetic clock, which is that, well, you can look at effectively the state of somebody's epigenome and come up with an inference of how aged that genome looks. And there are all the things that you might expect increase epigenetic age, like age and stress and so forth, do so, and the things that you might expect to reduce it, like exercise or reduction in stress, also do so. And so you actually have fairly good proxies for overall health as just a function of that. But the second part I'd say is exactly what Dr. Williams got at. It's really hard to target specific places to add methylation to. And if we think about cancer, it's been incredibly difficult to do so. And maybe one really good line of evidence that we're better off focusing on the demethylation or the loss of epigenetic features is from cancers themselves. The types of genes that get mutations in cancer are frequently those that go ahead and remove methylation or other epigenetic marks. And those ones get a whole lot of mutations. Their converse, the ones that put them on, don't actually get mutated very much. So it's as if saying it's easier to become a cancer by interfering with the taking away of the epigenome than it is to interfere with the putting back of it. And so I think that's part of why we see a lot of these studies and drugs focusing on that, because to some extent, the natural selection of the cancer is indicating to us that might be an easier pathway. That doesn't make it impossible. Everything in biology is possible in some way, but it's probably the case that we'll see drugs on that pathway much sooner.
And one last one. Yes, please go ahead.
So Dr. Scheer, I just wanted to clarify or refer back to your first slide because I'm not sure if I'm remembering this correctly. Did you say that only 20% of the families that you've tested had genetic factors for autism?
No.
No, okay.
So that's a good question. So, okay, so we think that so I'll backstep. If you study identical twins, so monozygotic twins that have the same genome, actually, the same genome sequence. If one child has autism, the second child, the identical twin, will also have some form of autism. It could look very similar. It could be somewhere on the autism spectrum, and that drops significantly if you're looking at dizygotic twins, and then it drops to the first cousins and general population. So these are, and then we see a recurrence risk in families of, if there's one child with autistic, there's an 18% chance of having a second. So these are the hallmarks that genetics is involved. So we tend to say that based on the monozygotic twin studies, 80 to 90% of autism has genetic factors involved. Okay. So then if we take, and we've done this with Christian, we've taken 100 consecutive diagnoses of autism that have come through our study, and we run the highest resolution genetic test, whole genome sequencing. And for 20% of those families, we could tell them what the major genetic factor is, i.e. the cause of the autism in that family. Okay? Now it's pretty conservative. We have a new study you'll hear about later this year that's going to add another 5% to that. And then if we look at 2 genetic factors together, it inches up a little bit. So I think we can explain probably closer to 40 or 50%. For the severe forms of autism, we actually can explain up to 50% now. These are so-called high-impact genetic changes. If you carry one of these, you're going to have some neurodevelopmental challenge, and often it looks like autism. But then, in addition, it's very heterogeneous, very complex. There's lots of what we call common genetic risk factors, the ones we've been talking about on the 23andMe chip array and things like this. So, so just to give you a quick, very, very quick biology 101. These rare genetic changes we see in that 20% of families, they don't get passed on because the kids who are autistic, they tend not to have their own kids. It's not that they can't biologically, they don't get put in a social situation where they pass on their sperm or their eggs, right? So those genetic variants don't get passed on, so they stay rare in the population. That's why so they're high impact, they don't get passed. But there's lots of common genetic variants that that contribute to your cognitive abilities, your speech and language. And we now know that there's probably thousands of these common genetic variants that make all of us unique in some way. I always say when I talk about our autism, it's a study of the human condition, because what's involved in autism is speech, language, cognition, and social interaction, our ability to socially interact with each other. This has been shaped by the evolution of our species. In fact, it's been the factors that have allowed us to be the most successful species, our ability to nurture and communicate and socialize and empathize. That's actually hardwired in the common genetic variants that are passed on from generation to generation. And we've in this polygenic risk score, there's a lot of data coming. In fact, we have another new study coming where we can attribute probably the remaining 70 to 80% to these common genetic variants. It's if you inherit a combination of, say, another 500 factors, if you have like the so-called perfect storm of these factors come together, then you can have the common genetic variation form of autism. It tends to be less severe. A lot of people have Asperger's, so this is what you learn more about. So anyways, for the 20%, it's these rare genetic variants that have a clinical impact that if you carry them, you'll have autism. And then for the rest, it looks like there's lots of hundreds of different genetic variants, plus environment involved.
I'll say in 2 words, everything, this isn't 2 words, everything that Steve just said holds true for cancer with just slightly different numbers.
Yeah.
Instead of 20%, it's more like 10 to 15. And instead of 50%, it's more like 35 to 40. But basically, that entire story could be replicated for oncology as well.
And I just want to really put a plug, this is more for the students and or parents of students. It's so important to take in your undergraduate year statistics. Again, so genetics is a science of statistics, math in particular, as high school and university. and the chemistries and the basic sciences as an undergraduate, because you don't get it afterwards, especially in professional school. And PhDs, you might get some 6-week courses, but you have to get it at this level. And the best graduate students and scientists have that training in their basic sciences. And we were talking about what we studied when we were here. If you're interested in science now, it's 50% of my staff sit on computers, and that increases. No one's doing experiments anymore. They're using big data sets. So we hire physicists and computer scientists. Actually, we hire co-op students every term here, and we have one laboratory and one computational science, but it's more and more computational sciences.
Yeah,
But statistics is the most important thing.
Yes.
Well, I know
Math.
that
They need to require grade
this-
12 math of all students,
Yeah.
which they don't.
I know we can go on for probably another hour, and I'm getting the signal from Joanne that we need to wrap it up. But there is going to be a reception. You can have some one-on-one time with our panelists. I do want to take this opportunity personally, and also behalf of the audience, to thank our panelists, 2 amazing alum from our university, and also one amazing professor who is educating our future alums. So thank you very much for being our panelists.